All right. Thanks very much, everyone, for joining us for our Vaegan seminar in term three, where we have Professor Li Zhaoping from Max Planck
Thank you.
Institute. She obtained her Bachelor of Science in Physics in 1984 from Fudan University, Shanghai, and her PhD in Physics in 1989 from Caltech. She was a postdoctoral researcher in Fermi National Laboratory in Bethlehem, Illinois, and Institute for Advanced Study in Princeton, New Jersey, and Rockefeller University in New York. Professor Lee has been a faculty member in computer science in Hong Kong University of Science Technology and was a visiting scientist in multiple academic institutions. She and her colleagues founded the Gatsby Computational Neuroscience Unit at the University College London, and from October 2018, she was appointed professor at the University of Tübingen and a Max Planck Fellow at the Max Planck Institute of Biological Cybernetics. Her research experience throughout the years ranges from areas in high energy physics to neurophysiology and marine biology, with most experience in understanding brain functions in vision, olfaction, and in non-linear neural dynamics. Tell us more about her exciting research. Please join me in welcoming Xiaoping.
Thank you so much for this organization. I really appreciate it. I also wonder if I could ask that you do turn on your video because this way it's actually I feel like I have, facial body language interaction such as, when I see you nodding or when I see you puzzled and confused, I feel like I can adjust in a more dynamic manner. And also I feel like I'm having a conversation. Is it okay? So if you or turn on your video. That will really help me. Thank you very much. And feel free to also, of course, have a drink with you. We are all like a group meeting. Everybody talk to each other. Yeah, and great. So let me share with this central peripheral dichotomy, and then wait, how come it's not? Okay, so you may ask, what is the theory? But in fact, I'm going to tell you this theory through its motivation. So it's kind of a history of why, and in the process, we'll say what the theory is. And then, of course, every theory needs to have experimental test. And this is, I hope, trying to make it stronger because the test can even falsify it or confirm at least the prediction, then further tests and so on. And so the motivation is we see that historically we actually only start to real have system vision science recording, system neuroscience recording in retina 1953. But immediately this technique is, getting this great leap forward achievement in V1 and finding these V1 neurons, seminal work, really impressive, founded a great foundation for us. But however, V2 seemed to be the beginning of difficulties because 10 years of work enabled us to know V1. Why is V2 taking us so many years and still we don't know as much as we know about V1? Yeah. And so this motivates the theoretical question. Every experiment to do actually is motivated by a theoretical question. Why do you want to do this experiment versus that experiment? So are we asking the right question? And in particular, you can even step back and say, what is vision? It's a big question, yeah? And so V1 actually prompts us to do that. And in particular, you say, what is the functional role of V1? And people may say, what do you mean? Hubel Wiesel already told us. But actually, what Hubel and Wiesel told us were only about the physiological role of individual V1 cells, not the functional role of V1. Okay, so that, just like we did not know the functional role of hippocampus until this accident of patient HM happened, and then we didn't realize it's actually for memory. And so therefore, What is the functional role V1? I like to go back to what is vision. Vision actually has a bottleneck, which we often study in psychology, such that we know that we can't see the gorilla in the midst of people playing basketball. But in fact, more than a century, half a century ago, we already knew there was a bottleneck. So somehow this was studied in the realm of psychology that we ask questions such as whether this bottleneck starts, before the object recognition of recognizing gorilla or after recognizing gorilla, but not quite into put the gorilla in the memory, or even before the 2D to 3D transformation, and so on. And this is called early selection or later selection by this bottleneck. And all these debates are in the functional domain, but not quite in the physiological domain where you say physiologically, anatomically, is it before V1, after V1, after V2? This has not been asked. Okay. In particular, whatever has gone through the bottleneck often is located near the focus of our gaze. And this gaze position is actually selected by your gaze shift. So if you started having your gaze in the center of this image, this flower is actually in the peripheral of your visual field. But once you have the gaze shift, it moves it to the center of the visual field. So it's as if you are moving a peripheral location into the bottleneck. Yeah. And this selection is a gaze shift. And then we can use this to say that vision actually, you can redefine it as both deciding where to shift your gaze, looking is actually a motor act, and seeing is a recognition. And we have been traditionally focusing on seeing as the view of vision is to finding what and where. But the idea is how do you know where to find is kind of less emphasized. And therefore, if you put this as one of the big elephant in the room for vision, then you completely, not exactly 100%, but it's a large portion of vision is this, tip of the iceberg, under the tip of the iceberg problem we have to address. And hopefully, by addressing this problem, you will understand beyond V1. This is the hope, but let's see whether we get it. Okay. And so, to address the question of where is the bottleneck, I like to motivate it by this theory I proposed more than 20 years ago, which is called V1 saliency hypothesis, which is actually addressing the functional role of V1. So V1 not only have neurons tuned to orientation and so on and so forth, it's actually building a saliency map, which is a map of the visual field full of scalar values. And in one particular location, it will have the highest scalar value, and other location are the second highest and third highest. So you have this scalar value of how salient a location is. And this map is then downloaded towards superior colliculus. And this anatomical connection between V1 and superior colliculus is monosynaptic and has been known for decades. But somehow, in retrospect, we now see what it could be, but it did not quite make us think that V1 is doing this functional output for movement. So superior colliculus can read this map and identify the highest saliency spot and then direct muscle movement to the spot to move a peripheral location into the central visual field. So you may wonder, say, oh, wait a minute, superior colliculus also listens to parietal cortex and frontal visual field, and frontal eye field, and so on. And they are doing the eye movement. And that's true. Traditionally, we think they do the eye movement. Yeah. However, I like to argue that they do only a small portion of the eye movement. It's like what they do is the tip of the iceberg. Because normally we actually move our eyes 180 times per minute. But we typically subjectively feel we only move our eyes 5 times or 10 times. That means we are feeling this voluntarily directed eye movement, and they are just the tip of the iceberg, and they are controlled by these higher brain areas. And you may also say, okay, if you say a lot of eye movements, lower visual areas, how about retina? Okay, Retina also sends input to superior corticus. Now, however, in primates, retina only send 10% of its neural axons to output axons to superior corticus. And we have experiment by Tadashi Isa and Yoshida. They have shown that if you lesion V1, then the monkeys can no longer do visually guided saccade before the brain reorganizes itself to achieve blind sight. That means in normal situation, the retina input alone cannot do visually guided saccade. Although the monkey can do memory guided saccade, that means the motor output hasn't changed. It's just a saliency map or some other map is not functioning. And so therefore, if you allow me, we say, okay, we can ignore these lesser players of eye movement and CV1 as one of the major players, then we should say this bottleneck should start right after V1. The reason is this eye movement is trying to select the fraction of visual input into the bottleneck. Yeah. And so therefore, if you have already done this selection of selecting where to move your gaze, why don't we start the bottleneck right after V1 rather than procrastinating into V2 and V2, V3, V4, right? For economic reasons, we should do it right away. And so this is a proposal of this central peripheral dicot. It's a very key proposal. And therefore, after V1, downstream from V1, we have to understand it. We have to understand it in light of this bottleneck. Without feeling this bottleneck, the question will be different. Yeah. So in particular, now you have impoverished visual information and such that you should have difficulty in seeing. Okay, we know we have this crowding and all these kind of things. We have been studying them as if they are just phenomena, but in fact, in the whole big field of a question of what is vision, this is actually a manifestation of bottleneck, makes seeing difficult. Okay, and in particular, this difficulty is also there in the central visual field, such that if you see this too briefly, let's say you see it for 10 milliseconds or so, you may not be able to tell whether this is a red flower or a red apple. And in such a case, central peripheral dichotomy theory actually proposed a hypothesis that central vision is specialized for seeing that it has a privilege which is not available in peripheral vision. That means it can go back to before the bottleneck to ask for more information. Say this information should distinguish the flower from the apple. Now, just imagine you only have like 1% of information available to feed through this bottleneck. And so you cannot just wait for the other 1%, for until 100% comes in, then you distinguish. You have to be selective to take the second 1%. So therefore, you need to query for this particular information that is useful to distinguish between flower and apple, not to query another irrelevant 1%. And so this is a kind of a problem specific on your current recognition. You say, is it flower or apple? So this is an online situation. And this particular query is the computation of analysis by synthesis. That means you're trying to analyze the difference, analyze whether it's a flower or apple by synthesizing that if it's a flower, which V1 cells respond in which way. And then you go back and query from these V1 cells, are you really responding in a way for the flower or in a way for the apple? So this is kind of, you have a model of the visual world, knowing which V1 cell should respond in which way for which visual input. And this is an old idea, analysis by synthesis. And what I have added new here is that this is only in central visual field. Okay, so this is the additional thing added by the central peripheral dichotomy. And so at this point, you may start to wonder that I'm building a sandcastle, okay? So many sandcastles. One is that bottleneck starts right after V1, and the second is that V1 is actually having this particular functional role to direct a gaze shift motor control, and that beyond V1, we don't see much. We have to understand it by this central peripheral dichotomy. And this sandcastle, you got to be suspicious. Okay, what is the background and what is the experimental data for it? And this sandcastle, of course, is built on a solid foundation of the primary visual cortex knowledge by Hubble and Weasel. And so let me go through these sandcastle to say that they are not really sandcastle. We do have good motivation for them. Layer one, layer two, layer three. So let's go to this layer one. Is it really true that we do have an attentional bottleneck starting right after V1? Okay, in particular, what information did not survive the bottleneck? Okay, this information is actually about the eye of origin of visual inputs, such that if we view something monocularly, you are not able to tell whether it's shown to the left eye or right eye unless you cheat by, closing one eye to check. Or unless there's some artifact, such as one of your eye is much weaker than the other eye, let's say. And however, V1 does have this information because V1 has many monocular cells, some tuned to the left eye, some tuned to the right eye. Of course, V1 also has binocular cells, but these monocular cells are so massive that you actually have this information. And this information is not available in V2. Largely, all of the, or most of the V2 neurons are binocular, such that V2 neurons activation could not tell you whether this activation is by left eye input or right eye input. And this could be mostly the basis for why we could not tell which eye give us the input. And so therefore, at least there's some basis for this bottleneck from V1. Of course, the bottleneck, and let's say is eventually, let's say bottleneck is only passing through 1% of the information. Now, from V1 to V2, you may only filter out 50%, let's say not completely 99%. So at least we started this question that bottleneck starts from V1, exactly how many information is filtered out is an empirical question, can motivate future study. And you can say, okay, now what is the next stage filter? Another, by V3, how much more is filtered out? Now this become a paradigm that you could ask this question further on, yeah. And so now I'm going to show you some basis for the second layer of this sandcastle, let's say. So the evidence actually, we are going to use this eye of origin information, which is only available in V1 and absent beyond V1, and use this information to show that it can actually direct your gaze shift and putting the strong evidence. Yeah. So the idea is if you make people do a visual search task to look for a uniquely oriented bar, people can find it right away. Usually within half a second, they press a button and found it. Unlike that, you could not see the gorilla in your midst. Somehow our gaze shift or attentional shift immediately enable us to see this target. Yeah, and you can even do that using a 3D goggle, which you don't have to use now, but later that people can still do it even if you put all the bars in one eye, no problem. However, if you make the stimulus a little bit different, to put a non-target eye into the other eye to a non-target bar, and then this unique bar, well, obviously you don't see it's unique because perceptually it looks the same, and nevertheless, V1 can tell it's unique, and this is what we observe in experiment. Okay, and then observers usually have their first, usually means actually most of the time. Okay, in our data, it's 70% of the trial, their first card goes there before 300 millisecond. Okay, and typically before 300 millisecond. And this is such a short time that you don't even have time to start binocular rivalry. Okay, and usually they have this first card and realize that this is not their target, and they do a second card and find their target, and they immediately press a button, and then you can ask them and say, look, ask you, this is a reaction time task, you should do it as quickly as possible, don't get distracted. But you get distracted. And observers typically say, no, 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 I did it immediately. I didn't get distracted and find a target and press button immediately. And they were not aware typically that they actually made a distracted saccade until you show them the gaze shift and then they say, oh, really? I did that? And so this is yet another example. It's like they're not aware. This is under the tip of the iceberg, reflexive gaze shift. And this is as if this task is heavily distracted by some, different colored bar, but this color is only visible to V1, and higher visual area could not see it. But V1 got so distracted, that's why they did that. Okay. And at the time when I originally did an experiment, that's 2008. I didn't do the eye tracking. I showed this by a reaction time, prolonging reaction time by this distracting by 200 milliseconds, really very long. And people find that this is unbelievable. Maybe I made a mistake in my experiment and so on. And so, why do people think so? This is an indication of our traditional concept. That our traditional concept just prevented us to think that this could happen. That means maybe our traditional question of what vision is, that vision has looking and seeing, and this traditional concept was not there. And that's why this motivated lots of people trying to repeat this experiment. And then its replication by many labs make people start to realize this is a critical element of vision. Yeah. And with this, then you may ask, what is the neural mechanism that make you do this shift? Well, actually, the neural mechanism has been studied as the surround suppression physiological stuff, because V1 neurons actually are recurrently connected by these short-range intracortical connections, and they such that neighboring V1 neuron suppress each other or excite each other. But one of these things is called isofeature suppression. Okay, in particular, one of them is called iso-orientation suppression, such that neurons tuned to the same orientation will suppress each other. So all these background bars activate neurons tuned to their orientation. They suppress each other. So this foreground bar to a different orientation escapes such suppression. And so therefore, the activation of V1 neuron is that this one is unsuppressed activation. And then if this activation map is your saliency map, and then you should have your gaze go to it very quickly. Yeah, and this is why your reaction time for this task is very short. And even when you look at molecular, not a problem. However, if you dichoptically put one bar in the other eye, you have another isofeature suppression. Is called eye of origin suppression. That is also known to exist in V1. That means V1 neurons tuned to the same eye will suppress each other, and tuned to different eyes suppress less, such that this unique eye of origin bar will escape such suppression, and therefore the activation map becomes that. And so this irrelevant bar will then attract your gaze. So, am I speaking too fast? This is the right speed. Okay. So, is it okay? Yeah. Thank you very much. So, therefore, this is motivating a following up physiological experiment with my collaborator Wu Li in Beijing Normal University and Yan Ying. And what we did was we make monkey fixate centrally first before we presented the bars, and the monkey should immediately try to find a uniquely oriented bar and succumb to it as quickly as possible. The monkey could not really predict whether this target will be here or there, so it's unpredictable. They have to find it. So therefore, you can imagine that if this bar is salient and evoke higher V1 responses, the monkey can succumb it to it faster than otherwise. And some of the times, this bar happen to be where we're recording simultaneously, and usually the monkey takes about 200 milliseconds or longer before succumbing. So therefore, you have a 200 millisecond window you can record this bar's response to this bars. So the recording response is the vertical axis and versus the time since the bars appeared. And you find that even this is the response to the same retina input interleaved with other trials when the target is not there and not exactly this kind of visual input. And so you pull out the retinal input, this exactly the same retinal input, and across these trials, you find that some responses are higher than others, even in the initial responses. And turns out that for trials with a faster saccade, the initial response is higher than trials with slower saccade. And so the same visual input gets to this behavioral fluctuation induced by the physiological fluctuation of the responses. This is as if then you are, let's say, imagine you can do optogenetic stimulation of this V1 cells to make it higher response or lower response, and then find behaviorally as a result, it has a faster saccade and slower saccade. But we didn't have this optogenetic technique. And so therefore, we just use spontaneous fluctuation of V1 neurons and correlate this fluctuation with the behavior. And this is the saliency signal, yeah? And this saliency signal happens so early, it's such a short latency that it is really hard to argue, even though at this moment we cannot rule it out 100%, that this signal comes from feedback from higher visual areas, such as frontal eye field. Yeah, so therefore, with this, you may, this really says that V1 is, not only we have this very surprising behavior result that really fingerprints for V1, we have this physiological correlate, and so V1's functional role, and this is the first clearest functional role we kind of go from behavioral, computational theory, physiology, and computational modeling, that V1 is actually a motor cortex. Yeah, so it's not only a sensory cortex, but also motor cortex. It's doing this action, and this can be done even without seeing. So this looking happens before the seeing. Looking is action, and you may argue, say, look, wait a minute, V1 doesn't quite synapse onto muscles, right? But motor cortex also do not have direct synapse, monosynaptic synapse onto muscles. Motor cortex needs spinal cord and brainstem to talk to the muscles. So therefore, V1 is no different in that case. Of course, V1 is specialized only for eye movement. It's not, controlling your limbs or, other things. Yeah. And so therefore, you can see that looking indeed can proceed and separate from seeing. And in fact, I like to also give you another demonstration, okay? And so we have a demo, and this demo is showing that they are indeed separate processes. In our everyday visual behavior, you may feel they simultaneously happen, they must be the same thing, but no. So the demonstration use again this very simple case of looking for a uniquely oriented bar, but you see that this is easy. But if I superpose on each of these bars a horizontal or vertical bar, then suddenly you find it difficult, yeah? But V1 is responding to all of these bars, yeah? So no problem. And but IT is seeing the recognition of the X shape. So this is something I did not ask you to do, but your IT automatically does it. And so IT is doing the seeing of recognizing the X shape. V1 could direct looking by, say, because this is the only bar, the target bar is the only one that is evoking the highest V1 response. And so therefore V1 can make you look, but IT will make you see and confuse you. Okay, and because IT see this X is indifferent from other X, even though they are rotated by 90 degree, 180 degree, recognition is rotationally invariant. As you can see that if you make this X a bit skinnier, you don't get so confused. Yeah, and so indeed it is the this kind of a rotational invariance. And now I'm going to give you a demo with a huge expanded bigger version of this input. And I'd like you to immediately look for this uniquely oriented bar. If you see it, find it, immediately raise your hand. See if you can do it. Yeah. Okay. Ready? Trust your V1. Yeah. Ready, steady, go. Find the uniquely oriented bar. Great. Michael, find it. Okay. Juno, find it. Okay, Kenzo's struggling, Peter's struggling, yeah. And so it's amazing that you could not find it so easily, but in fact, it's right there. And if you start your gaze not near it, let's say typically you start your gaze in the center of the visual field, you will not be able to see it because you're crowding, okay? It's in your peripheral visual field, is in crowding. And so you have 1,000, more than 1,000 bars. How do you know to shift your gaze there, yeah? But this is a typical gaze shift from a typical untrained subject because my subjects never been trained on this, and this is their initial trials. Okay, so within 1 second, they succumb to it. And how often does this happen? It happens in 50% of trial. Their gaze go to it within 1 second. This is amazing. So therefore, after 2 seconds, 70% of your trials have already gaze shift to it. And within 3 seconds, almost 90% of trials. So if I wait 5 seconds, you still haven't raised your hand, this is because your seeing start to happen. Because once your gaze go to it, now it's in your central visual field. And your central visual field immediately make you see. But before your gaze went to it, you did not see. So you look before you see. Okay, and then your seeing happens, and the gaze shift afterwards is plotted in this red traces. And the seeing is, first of all, you do a bit hesitation around it. You are kind of you know, what's going on. You see it, get confused, and then you decided that, okay, that's not a target. You keep looking, yeah, keep searching for it. And so you see that looking and seeing are indeed separate, and this is a pathological design to see the separation. And the looking is by peripheral vision, and seeing is by central visual field separately. Okay, is this okay? I like to. Okay, fantastic. And so now I kind of I even did an experiment with a macular degenerate patient that doesn't have central visual functions. And she says that, oh, well, she is just guessing. Actually, I make her do this task in the touch screen. I just say, go touch the location of the target. And every time she touches, she says, oh, I'm just guessing. I'm just guessing. And she didn't get confused. And she does very fast. You know, this just shows that without this central vision, she doesn't get confused. Yeah, she could not see. She could look. And now we are ready to talk about the seeing and in light of this bottleneck. In particular, of course, we know it's very difficult in the peripheral visual field. This is the traditional, you know, very well-established phenomenon of crowding that in your peripheral vision, if you don't have enough information, you could see, you have enough information in the retina because you can see the T to the left of fixation, but not to the right of fixation. They have the same retinal information. Yeah, retinally around the T, all the pixels, obviously, information sufficient for you to see it. But nevertheless, if you can overcome this crowding by putting the central visual field, this is because you can go back and fetch for more information to distinguish whether it's flower or apple, or distinguish whether it's a T or not a T. And so central visual field has this privilege, peripheral information doesn't. And rather than using crowding, I'm going to use something a bit more informative, which is illusion. So crowding is you have ambiguity in seeing because of impoverished information. Illusion is you make a mistake in seeing because of impoverished information. So this predicts that this illusion is more visible in peripheral visual field, not in central visual field. So even though peripheral visual field you should see less, but in fact, illusion you should see more because you don't have a mechanism to query for more information and overcome this mistake. So for example, let's see this particular example. You see that, wow, these, you know, illusory motion, even though this is a static image. But do you notice that wherever you stare at the center of your fixation, this illusion is not there, right? And so this illusion is only in your peripheral visual field, just a few degrees away, it's there. And this is because this illusion is vetoed in your central visual field using the additional information queried by the feedback query. Okay? And so therefore, you may say, well, look, wait a minute, we already knew this illusion, yeah? And so this is consistent with your theory, but if your theory is correct, can you predict new illusions? And let's see what that happens. And this is what we're going to do. We're going to predict new illusions, okay? And this new illusion is quite special in the sense that most in your vision science field community, illusions, we happen to notice them by accident, not quite predicted by theory, but this is one actually predicted by theory. So this illusion is the perception of tilt. And this is in this image, you see lots of things tilted to the left. This is not an illusion. This is a baseline, okay? In the baseline situation, you see lots of these pairs of dots. The alignment between the two dots in the pair is tilted to the left, okay? So you see this tilted to the left. And in this image, it's also everything tilted to the left, except the pair of dots is one black dot, one white dot, okay? It's always different colors. Here, the pair of dots is always the same color, a pair of white dots, a pair of black dots. And so this one, you see it's tilted to the left. But if you fixate your gaze here, you see that it's tilted to the right, the opposite tilt. Can you tell? So fixate your gaze here and trying to see the tilt there. It's actually the opposite tilt, yeah? So if I make you to force choice in a psychophysics experiment, you will actually say most of the time it's tilted to the other way. But it's difficult in this demo, but let me demo it to you, okay? Let me first of all see how I derived it, how I predicted. So imagine you have a pair of dots. It's actually going to activate a V1 cell. We know this is a Gabor filter tilted in its orientation. No problem. But it could not be activated by this pair of dots, yeah? And because the black and white dots cancel each other in this off subfield of this Gabor filter, but it could activate another V1 cell tilted to the other direction. Yeah, and so that means this V1 cell is fooled by this input into thinking the other orientation. Well, in V1 we have 100 million neurons, so you fool one neuron, you cannot fool the whole brain, unless this is the only neuron that goes through the bottleneck. Okay, other neurons did not go through the bottleneck. Okay, and so this bottleneck is the icing on the top, based on this traditional information provided to us by Hugo Weasel. So the icing on the top is there's a bottleneck, and this most activated neuron by this is the one going through the bottleneck. Of course, if the higher visual area can go back and query for more information. And what information could it query? Well, it could query, say, well, I know this cell is cell number 13, and it's a little bit, sometimes can be fooled. And so you say, oh, let me look at cell number 14 that's having this residue field, and it doesn't get fooled, right? Okay, or look up the cell number 15, or look up cell numbers 17 or so. So if we want to look up 3 additional neurons responses, then it can correct its confusion. Yeah. Then, and this lookup central visual privilege is the way to overcome being fooled by the most activated neuron. Yeah. And this is why you will have, this is why I predict this illusion. The illusion is because of the bottleneck when you cannot look up, okay? And so this is predicted. And so therefore this original input going forward, and then you look up for this cell, this cell, this cell's response. And that means your higher brain area has a model of the visual world and how this cell should respond if this is actually this input. And you look up, oh, V1, is this cell indeed respond this way? And if so, then it should be this input. Yeah, this is the idea. So let's show this illusion in a demo. And here you still don't need the 3D glasses yet. In this demo, you see that you have lots of orientation along the tangent of a circle, no problem. But here is the same circle in which every second element I make the orientation perpendicular to the tangent. And then such that if you put in a noise, this circle is less visible than that circle because every second one is perpendicular, okay? And now I can demonstrate to you the illusion that if you have the illusion, now every second I make this hetero pair of dots. So if you have the illusion to see as perpendicular, then you will also have a problem of seeing this circle in a noise. Yeah, this is the demonstration of the illusion. So let's see. Even though in your central vision you see it's parallel to the tangent, but if you look the whole thing in your peripheral vision, you cannot see the circle, right? Okay, so that shows that in your peripheral vision, if you're fixated here, they are all in your peripheral visual field. You can't see the circle. You fixate here, no problem. Yeah, and although if you look in the central visual field, this looks perfectly smooth. But even if you look at central visual field, there's other segments of the circle in your peripheral visual field. So that's why, yeah, it's difficult. And just to convince you a bit more, I'm having another situation where I actually having these elements perpendicular. But in your peripheral vision, you see it as parallel, yeah? So you in your peripheral vision, you see these elements as parallel to the tangent. And so therefore, if I put this in the noise, you should be able to see this more easily, this circle more easily than that circle. Okay? If your illusion works. Okay, so let's see. Yeah, which circle is more visible? To the left or to the right? Yeah, so this shows that this is a demo, this illusion works. And even though in central vision you see this is much more smooth than that. Yeah, so therefore your central vision doesn't see this illusion, your peripheral vision does. And now you may immediately, it should be critical to say that, wait a minute, this is such a complicated theory. Can't we have something simpler? Alternative theory, alternative explanation? That central vision has higher acuity, so therefore it is less vulnerable to such misleading information. Okay, and so this theory is simpler when we have the same phenomena explained by two theory. One is simpler, we should prefer the simple one. So therefore, to distinguish between these, between the alternative and my theory, we need to give a prediction that can rule out the alternative. And so another prediction is that if you cut the feedback, okay, now you should be able to see it even in central visual field. The central visual field should be able to see this illusion. So how do I cut the feedback? Yeah, so I do not have the ethical approval to have the subject come to my lab, and then I open their brain and cut the feedback. And so therefore, again, we have this nice treasure in psychophysics toolbox. Where I just cut the feedback backward masking. Okay, so the idea is that make you see this image for a very short time. And so let's say if you see this image for, present the image for a short time, you have a feed forward information. And by the time you feedback to query for additional information, it's already a mask, no information available. Yeah. And so this is the idea. So I let you show it for 10 millisecond, 20 millisecond, and so on. And sometimes I make you view it for 100 millisecond, such that the feedback has already happened. Sometimes I make it too short. Okay. And then I also do it for the baseline situation where the feed forward information is not misleading. And then you see that this is their accuracy in performance versus the time presented in the baseline situation. So if you make them see long enough, their accuracy is 100%. And their chance level should be 50%. Yeah, if their task is to say whether the whole thing is tilted to the left or right. Okay, so two alternative false choice. However, as you get shorter and shorter, you're pushing towards their temporal resolution limit. But nevertheless, it's still better than chance, even at 10 millisecond. Yeah, and so this is good. Now, if you do the test situation, these trials are actually interleaved in the experiment. And you find that, yes, when it's long enough, when the feedback is available, they do incorrectly. But when the feedback is less and less available, this is significantly below chance. And this is the illusion. Okay. And so therefore, we have this prediction showing that. Is this good? Should I pause a little bit? Is this good? Yeah, great. And now you will have a chance to, I'm going to extend it to reverse depth illusion where we will have this anagram. And because this reverse depth illusion is even more indicative of V1 because they're analogous, just that flip tilt illusion. The idea is if you have a random dot stereogram, you have this, let's say a disk in front of a ring, you will see two images to the two eyes slightly different from each other. Okay, this is the actual image we show to the subjects. And every black dot in one eye corresponds to a black dot in the other eye, a white dot in one eye to the other eye. And so a V1 binocular neuron tuned to these two dots will say, well, this is my disparity, and my disparity preferred is near disparity, so I'm excited. And so all these near disparity tuned V1 neuron will say, this is a near depth. This disk is in front of the ring. Okay, this is a baseline, no problem. And let's just establish the baseline situation with your N graph. And if you have your N graph, you have it with the, so let me put it a bit brighter. Okay, so how do I do it brighter?
All right, it's
Yeah,
a
so
red lens to the right,
yeah,
the
you put
left eye.
the blue lens. Let's say you have the blue lens in front of your. If you have a blue lens in front of the right eye, you should see a disc in front of the ring. Yeah. If your red lens is in front of your right eye, then you'll see the disc is behind the ring. And so anyway, let's say you see it. Is it okay? Everybody see it with the lenses. Yeah. If you see it, give me a thumb up. That's what I just say. Yeah. No? Yes, no. Great. Okay, fantastic. Okay, now I'm going to, this is the baseline. Now I'm going to, do you know this time, could you see it? Because last time you maybe, in a poor demonstration, in a poor,
So left red, I see center popping out.
okay, so you see center point, great. So this is the baseline. Cancel is okay. Yeah, great. Okay. So, and now I give you the situation where the illusion happens. Just like, your pair of dots, I now make a pair of dots, because I copy the left eye image and make the right eye image such that for every disk dot, I reverse the polarity. Black become white, white become black without changing position. So now the disparity of this pair of dots is still the same as previously, but the original neuron that's tuned to near is no longer being activated by it. Just like the flip-tilde illusion, the orientation has to flip 90 degrees for the neuron to be activated. And this is analogously V1 neuron tuned to depths. The neuron responding to this pair of dots is now tuned to far depths, not near depths. And this is physiologically observed. So that means V1 neuron is reporting to higher visual areas that this disk is behind the ring. Okay, so you may say, okay, would the higher visual area, brain see it as the disk behind the ring? And this was actually a VSS demo where people were shown these random dot stereograms, maybe motivated by this experimental finding, and say, what your perceptions see it as a disk behind the ring? And everybody in that demo said could not see it behind or in front. And that was, I was actually one of those in the audience. And it was amazing. And at that time, it was explained that because V1 is not where consciousness is. Okay. And, but now I have an alternative explanation. This is because these neurons feed forward saying that, okay, the disk is behind the ring. And the higher visual area say, well, it's a bit ambiguous. Okay, let me go check. And what kind of things could it check? It could check with a monocular neuron saying that, okay, this is a black dot because this monocular neuron did not feed the information to V2, right? And so it says check with the monocular neuron, this is a black dot. Okay, check with another monocular neuron says this is a white dot. And so therefore, these two monocular neurons are vetoing the information about sent forward by this binocular neuron, yeah? And so when the information about the disk behind the ring is vetoed, then you would not see the disk as behind the ring. Okay, so now let's see it. Put on your glasses again. And even though in the top you see the disk in front of the ring, here you see, well, center is kind of weird, in front or behind, I'm not sure, kind of thing. Yeah, so this is exactly what our VSS demo is saying. But now, if we say, what if you view this disk in peripheral vision? Okay, now peripheral vision doesn't have the ability to go back and check, and then you should see it as whatever V1 you want to tell you. Because that's a prediction that's been confirmed in 2018 by Zhaoping and Ackerman. So let's see it. Okay, now if that's confirmed, you say, well, it's not because V1 is not consciousness. The new explanation is central vision does not get fooled because it has the feedback query, but peripheral vision will get fooled. So let's try it again. You look at this stereogram, but don't look at it in central vision, okay? Trying to fixate on my mouse, on my hand, or somewhere else. Then you see that this is the central disk is behind. Okay, observers to my experiment. they say, oh, I don't know. I make them do forced choice and do 100 trials and so on. And then you do the statistics. And normally in my seminar, when I ask people to, raise their hand if they see one way or another, typical subjects see this as behind by 3 to 1 chance level. Yeah. Or 2 to 1. The twice as often to say this behind than in front. So this is, I wonder if you see it this way. Yeah, do you?
Yeah, absolutely.
Yeah, thank you. So this is really the benefit of Zoom, where you can see it on the comfort of a very good display. You can just brighten it. Michael, do you see it? Yeah, 50-50. So I need to force you to do 100 trials, first choice. Yeah, something like that. Yeah, so this is a subject often say they are guessing, but they often do it quite well. Yeah. And then now you do exactly the same thing. I say you will be able to see this illusion in your central visual field if I do backward masking. Then you say, do you mean you are going to let me see this random dot theorem for 10 milliseconds? I can't even fuse in 10 milliseconds, right? And so rather than make you to see 10 milliseconds, I make it dynamic. So every 10 milliseconds, I refresh the random dots. So it's the same disk, same range, same disparity, same binocular correspondence, whether it's correlated correspondence or contrast reverse correlation. Okay. Now, every frame of this, every 10 milliseconds, you have a new fresh set of random dots. The feed-forward information from these random dots to say the disk is behind a ring or in front of a ring is exactly the same. But you just can't do the feedback. Because if you want to go feedback and look at this dot, is it a black or white? It's a next set of random dots. The dot has already changed. Okay, so that's the idea. So let's look at dynamically. Okay, again, if I do dynamic flickering, so I just refresh the random dots here, of course, my laptop is slow. Maybe the Zoom is also slow, but it doesn't prevent you from seeing the disk is in front of ring here. No problem. Yeah. But now here, if you do not see in the second one, in this case, you just now look at in your central visual field. You don't have to look away. Okay, in your central visual field, you say, I cannot tell whether it's behind or in front. But now I'm going to flip it. You just look. And when you flicker, you should be better able to see. Yeah, although this is a little bit slow. So if you come to my lab, I can have a faster flickering in 10 milliseconds. And so people are better able to see. Does that work for you?
Yeah, I think so. Can you flicker the top one again?
Okay, flicker the top one. Okay. Okay, top
Yeah,
one, you
I
should,
think that's a good
the flickering
comparison.
does not prevent
Yeah.
you. The bottom one, flickering suddenly make you able to see. So if I don't flicker, you would not see. And so this is actually the data. In our experiment, I make people see it for 200 milliseconds and then block it, for example, mask it, but 200 milliseconds enough for their feedback. So you can have the true depths, you can have a fake depths, you can have the true depths, and you can have a fake depths. And so if it's true depths, whether you flicker, or not flicker, so whether the feedback is working or feedback is not working, subject to more or less 100% correct, no problem. But if you have fake depths, if you don't flicker, so their feedback is working, they are chance level, okay, 50-50. But if you flicker, they are so much worse than chance. That means they see the opposite than a true depths, yeah? And so the idea is they do have, it's quite very significant. And you may ask, okay, this is 10 millisecond, but what about 20 millisecond? Yeah, you want to know where the feedback comes in 20 millisecond or so. So that we did another experiment where you could have 10 millisecond or 20 millisecond or 40 millisecond, 50 millisecond, and 100 millisecond. And you can see that you say, oh, does feedback come, it's not like a step function, like, okay, now you cannot see, and suddenly you can see. It's not true. Okay, so they're slowly going down. It's as if that with faster blocking, you're blocking more and more feedback. This suggests that feedback may be coming from multiple brain areas, possible, or the feedback is actually going in iterations. Yeah, and so this really means that we should use physiological means and maybe other means to really work it out. But this is a Yeah. But anyway, we also did this peripheral vision, repeated in peripheral vision. You see, in peripheral vision, you don't need to flicker. You already see the illusion. And however, peripherally, we did it in like 7 degrees, maybe not peripheral enough. So when you figure, you still, get to be better. That means you probably eliminate more feedbacks. Yeah. And so therefore, this experiment is worth doing in further peripheral. You should see that no difference whether you flicker or not. Or more closer to the central, then you should get a bit, more slanted. So this is for future experiments. And so you see that this framework actually give you really, really non-trivial predictions. And these are all very surprising and they have been confirmed. And this is kind of a computational mean can ask very, very non-trivial questions and falsifiable. And so far, this framework has not yet been falsified. That doesn't mean this framework is correct because we have to keep on having more predictions to falsify it. Yeah. And so just that this is so far so good. And one particular prediction is actually this anatomical prediction that feedback is only privileged in central visual field. And so far we only have indirect, so far, I mean, before a couple of months ago, we only have indirect experimental support in terms of functional connectivities between V1 and frontal brain areas. And they found that indeed it's more central than peripheral. But functional connectivity in fMRI is hard to say. It's feed-forward or feedback and maybe even polysynaptic. Yeah, so it's not quite direct. And then we also have these analysis of V1, V4 data in Utah arrays and when monkey is in the black spontaneous activity by analyzing grandeur causality. And grandeur causality is not completely convincing to many people. So you may say it's, well, we need a bit more direct evidence. And this motivated my collaboration with Marcella Rosa and Pietro Maker, where Marcella Rosa injected retrograde tracing into V1 of marmoset monkeys. And this is really fresh data, very exciting to me because as a theorist, you say you're predicting this, you know that if it's by chance you're the 50%, you could be vetoed and 50% not vetoed. And so you inject retrograde tracing in V1 and then you let monkey live for another 2 weeks or so, then the dye will be transported to other brain areas where it's the source of the input towards V1. Okay. And so you get V2, V3, and V4. You can also trace back to LGM, which give feed-forward input. And so therefore, if you compare feed-forward input from LGM versus feedback input to V2, you can say how many V2 feedback per feed-forward, and that's a ratio, yeah? And so this is the ratio. We plot the ratio between feedback to feed-forward. Feed-forward is LGM. So for instance, V2 to LGM is this blue line. You see it? This is 10 injections, okay? One injection at this is eccentricity. So one injection at eccentricity 2.5 degree, another 3 something, and 4 degree, and 5 degrees, so all the way to 15 degrees. And you can see that it really does drop down. And even though data is noisy, you can say that because this is log scale. So from 2 degrees to 15 degrees, it's a factor of 10 difference. And this is quite amazing because you can say that, in a Van Essen diagram, when you have interconnected visual areas, V1 is a huge block. It's just one block connected to V2, V3, V4. We never think of V1 as two different brain areas, yeah? But in fact, you can say that central V1 and peripheral V1 are acting like two different areas, okay? So for instance, you can also look at this orange curve. This orange curve is actually for all the ventral string input. You just add up all these visual areas that belongs to ventral string, and then you see they also go like that. Okay, and you can also add up all these areas to dorsal string. This is this green curve. Okay, you can even have this ratio between ventral and dorsal string. So you can see that it's as if in V1, central V1, and mainly the feedback coming from the ventral string. In peripheral V1, the feedback mainly coming from dorsal string. In fact, this is also one of the prediction that Peripheral vision is for looking. You need a dorsal feedback for looking. And central vision is for seeing. Yeah, you need a ventral feedback is for seeing. So again, it's central peripheral dichotomy. So this is really exciting that from computation to anatomy to behavior, yeah, and theory to that. And the peripheral vision mainly for looking, central vision is for seeing, and central vision is for the having the privilege of the feedback. And this is motivated by a bottleneck, but all animals have bottleneck. So they all have to select a fraction of sensory input into their bottleneck for recognition. And while all central input is coming from central vision, a rodent central may be coming from the nose, okay? And so the rodent central is actually olfactory and microvibrissa, okay? And their vision is their peripheral. So you can take away their vision, they don't mind it so much as we humans do, yeah. And so there's lots of rodent vision studies, fantastic. However, conceptually, we should not confuse this, should not equate it to the primate vision because primates vision having a central and peripheral functions depending on, but rodent vision is more or less a peripheral function, is to direct attention. Okay, that's very important. And you may also think about another species that is an artificial species. Well, it doesn't have a bottleneck. Well, because, for instance, we humans, our brain is running on a limited power of 20 watts. This robot can be kilowatts, power, and we have a limited working memory capacity and so on. They can have unlimited, you can just put a lot of memory in there. They can remember the whole internet content, yeah. So no bottleneck. Now remember that all of our illusions were uh, predicted because of the bottleneck. And then you say, well, they have illusions? If they don't have bottleneck, they should not have illusions, yeah? Well, current robots, despite not having bottleneck, they are easily fooled. That means we are not utilizing the computational resources in their feed-forward neural network for. So this just says that our current AI algorithm, despite not having the bottleneck, is not good enough. So we humans have this feedback query is very hallmark of human understanding. The idea is when you feedback, you are actually using your internal brain, internal model of the world. You say, oh, I think V1 neuron should respond to the flower this way or that way. You are thinking, you have a generative model, can imagine how the V1 neuron should respond if it's a flower versus an apple. And so you say, I imagine how this should be this way. So you can say, I imagine if I do this move, you will defend by that move. And you can then I'm going to follow up to the third move. And then you will, you can imagine several steps ahead. Yeah. And so this imagination is the hallmark of human understanding. And AI, sometimes, you don't know why the AI recommend, doing surgery or not doing surgery. It's like a non-explainable AI. This is one of the things that we are faced with a challenge, to make AI explainable. This understanding is lacking. So therefore, I am now, as a theorist, I'm thinking ahead that this feedback imagination, that you can go in loops, can be very complicated. It can be metaphorical thinking that, oh, if I do this, what if that, and then what? Yeah, and then you can loop it so long that you can tell a whole story. And this story making and language, once you have uh, uh, sufficient this feedback capability, which doesn't require a lot of the higher brain areas, which could be your frontal brain areas. We humans have such a huge frontal that this is it the reason why we have language and other animals not having language? No, this is a very good question. Yeah. And so this is the idea that if we understand V1, it is hopefully enable us to understand vision. And so far we have understood some of it already. This is kind of a new question and new paradigms such as illusions, but I hope that this will lead to we have enough behavioral paradigm. Now you can follow up with physiological experiments. You can make monkey do this task of this disc in front or back. It's easy to train a monkey for this task while you simultaneously record from V1, V2, V3, and that will really So we not only have predictions about the anatomical prediction that will look and enable us to ask these questions in a more uh, different manner than previously we have been doing. Because previously, experiment V2, V3, you are not thinking about the bottleneck. Here, we need to understand V2, V3 before. In light of the bottleneck, with the idea that it should also feedback in which way and so on. And hopefully it will help us to understand our whole brain because our brain, actually do imagination, all these things. So this is the idea of this talk. So thank you very much.
Thank you very much. Do you have much time for questions?
Yes, of course.
Oh,
It'll
great.
be great. Yeah.
Questions. Kenzo, anyone
Yeah.
who's unmuted? Yeah.
Hello, Kenzo. Yeah, please.
Hi. I think that your central peripheral dichotomy idea can be supported by other illusion. For example, the scintillating grid illusion or
Indeed.
extinction illusion by Jack Nino.
That's
Both
right.
of them, we can see that illusion only in peripheral, but
Yeah.
not in the central vision. So, do you have any idea to explain those illusions by your theory and
Yes, yes.
mechanism
In
that
fact,
you, yeah.
indeed. So in this central proof dichotomy, in this 2019 paper, I even propose that if you have a particular visual phenomena that's only available to peripheral visual field, then it suggests this is because you lack feedback. But if you
Mm,
have phenomena that's only available in central visual field, then it must be involving feedback. So, for example, backward masking is much, much more effective in your central visual field.
mhm.
Okay, or maybe I should play such that the sharing can continue. Let me go back to sharing. Okay, sharing screen. This, yeah, screen sharing. Now, can you see my screen again?
Not
Oh,
yet.
not yet. Okay. So, here, share, and then you can see it again. Yeah. Great. Okay, so this is a proposal. So, of course, this can also act as a test of the theory. So you say, now I don't know, can I explain this scintillate illusion by your theory or not? Yeah, so indeed, you're right. Scintillating grid illusion is much, much stronger in peripheral vision and central vision. And then I predict this is because it is only using the feed forward and not feedback. And then people use that to study to show whether this is the case. If it's not the case, my theory is falsified. So I am actually by saying that. So for example, I'm trying to understand why is this rotating snake working? I think Michael Bach has some explanation about why this is working, and then, it is actually a little bit related to the reverse phi motion illusion. Now, reverse phi
Oh,
motion illusion is actually related to my reverse depth illusion and reverse
mm,
tilt illusion because again, it's flipping contrast. Yeah. And so
yeah.
in this sense, you again see that almost this rotating snake is actually an analogous of a reverse, basically reverse feature illusion. So you can have a
Mm,
reverse feature in orientation, reverse feature in depth. You can have a reverse feature in motion direction. And so that works. But scintillating motion illusion, scintillating dot illusion, actually, I do not understand. Maybe Michael has a way to understand why is that. And so therefore, this is motivating by this framework to actually understand it. Is it really because of lack of feedback? If it
mm,
is, then another confirmation evidence to my framework. If it is not, it's a falsifying evidence. Yeah, thank you, Kenzo. Maybe you have some of these other, these recommendations and additional other ones will be very useful
yeah,
for
yeah. Okay.
testing. Yeah, this framework. Thank you. Yeah,
Yeah, the examples really do help. Thanks,
yeah,
Kenzo.
yeah.
Michael, I think you had a question, but maybe you can also provide some perspective on the scintillating dot illusion.
Uh, well, first, thank you, Ali. This was a very inspiring talk. Totally
Thank you.
interesting. Thank you very much. And I learned a lot. Now, And I think the idea to use the flicker approach to probe the timing with respect to feedback occurring or not is a great idea.
Thank you.
That having said, you mentioned acuity across fields, or
Yeah.
what could also in this context call it M scaling.
Yeah.
And I think you played this down a little too much. I think every experiment you use where you compare periphery and so should be M scaled, especially the rotating snakes and the scintillating grid and so. And I even wonder if the tracing injections are also affected by this, because I think the peripheral central comparisons in tracer injection should be scaled by the upwards propagation.
That's why it's the ratio.
Yeah.
Okay, the
So,
ratio
but
is it's not just
up-down?
V2. No. So what we do
No,
is
I mean from LGN. That
yeah,
wasn't
yes.
in that experiment. Yeah, so the
We
ratio
do
is
have
not.
the LGN as well.
Yeah,
We
and
also
how
count the
does that
LGN.
behave with eccentricity?
We do the ratio. Okay, so the idea is, let's say we count the V2 has, the idea is if you inject, sometimes you may inject more, sometimes you may inject less. And so therefore, if you inject more, you will have more counts in V2. Inject less, you will have fewer counts in V2.
Yeah, yes.
And so we also go count the LGNs. Okay. So therefore, if V2 is more, LGN will be also more. Then we do the ratio. The ratio we say, okay, V2 count is 200, LGN is 10. And then we get this ratio. And then you go down, and you find this ratio go down. So the idea is it's already scaled by feed forward. So this is already M scaled.
I meant the LGN V1 feed forward for
Yeah.
scaling denominator.
LGN V1 forward is so imagine, okay.
But that's not in the experiment, is it?
It
The
is. In
experiment
the experiment,
doesn't
we also
add.
did LGN to V1. Yeah, we did.
Yeah. Did you use that as denominator for scaling? The LGN V1 forwarding, because that's M scaling, or that would possibly cover M scaling.
Yes. So maybe I should draw a little bit. Imagine LGN to V1, you find, so let's do the raw data, okay? Imagine you have a raw data. I'm going to plot the raw data count between LGN. So imagine you count raw data, okay, versus eccentricity. Okay, you find that LGN to V1 goes down. Okay, this is what you find. Okay, and you also find that, so this is LGN to V1. Let me make it cyan. Okay, you find that this is the M scaling. Okay, LGN to V1, but then you also find that V2 to LGN also go down.
And you made that steeper now. Does that reflect the
Steeper,
data?
exactly. It's steeper. And then I do the ratio between V2 to LGN. That's the ratio that's plotted. Okay, so the raw data is this way. But of course, raw data, this is hypothetical raw data. Okay, so let's say this is the LGN to V1. Okay, and raw data, of course, they actually very noisy. Depending, let's say we have one injection. Let's say one injection is here. Okay, one injection is at this eccentricity. In this eccentricity, let's say you put so much dye in it because you inject, sometimes it's more coming to the syringe and something. And so therefore, this injection, you may have a data point here for your LGN count, okay? And another data point there for your V2 count, okay? But another injection, you inject so much more. LGN count is actually here, okay? And V2 count is there. Do you see what I mean?
Yeah, of course, that's noise. That's clear. I mean, if you say
Yeah.
that these two eccentricity dependencies
Yeah.
have very different slope, then you're making
That's
your point of feedback being stronger in central
That's
than
right. We
peripheral.
take the
Okay,
ratio.
yes,
That's
that
why.
is.
Yeah.
Yeah,
Yeah.
because
This
in
is
all
idea.
the
Yeah.
legends in your figure, they show the other direction, not what's the LGN V1, but V1 LGN. Okay, but
Yeah.
if
So make
this
it
is
bigger.
what you
Thank you.
did, oh,
This
you
is a
said,
good
yeah,
idea
yeah,
to expand it bigger.
okay,
Yeah. And so
yeah,
let
yeah.
me take this away. Yeah.
Mm.
It's
No, okay,
fantastic.
fine,
Yeah.
yeah.
Yeah. So
Yeah,
you
but
said V2 to LGN, and so they're all scaled by LGN. Yeah. And then you say total feedback to feed forward. Feed forward is the LGN. Feedback is total. That means that regardless of brain area, this is this particular
okay,
curve.
yeah, so it's fine. Just
Thank
as a
you.
comment, the nice demonstration of the flickering approach don't really work well over Zoom. My frame rate here, which Zoom can tell you, was 12 hertz. So that would be,
Oh, that's
yes,
why.
exactly,
Yeah, 12 hertz is
Yes, I
like
mean,
a,
you can check yourself the frame rate in Zoom, and at 12
yeah.
hertz, of
So
course, it
12 hertz,
doesn't
it will
work.
be
You're at
this
the
one.
left end.
Yeah.
Yes,
Thank
that's
you.
just
Thank you. So,
so
yeah.
that was a feedback.
Yeah, so your feedback was actually 100 millisecond, 12 hertz. Well, 12 hertz like a 80 millisecond. And so therefore, you see, 50 millisecond,
Yeah,
we barely get
yeah,
significance.
okay,
So therefore, it may
okay,
not work
yeah,
very well.
yeah.
So I
But
would
still,
welcome you,
I would.
Michael. You come to my lab. I actually give you a demo. Yeah, that would be give us a
No,
good
in
idea
fact,
to have
I
a
when
cup
I
of
saw
beer
your
together.
paper, when I saw your paper, I programmed this myself. So I think it's there. It's weak for me.
Thank you. Thank you. Yeah.
Uh,
Yeah.
uh,
Yeah.
but
Yeah.
still, I would hold that everything should be M-scaled, and then other things on top of it.
That's right. That's right. Everything M scaled. And despite that, given the M scaling, you have a feedback to central visual. That's the idea.
Exactly.
Because this
That makes
feedback
much more
is
your point.
per already M
Yeah,
scale
yeah,
because
yeah,
it's per millimeter of
yeah, yeah.
visual cortex. And this per millimeter visual cortex is already M scaled. Yeah, that's the idea. Indeed. Yeah. In fact,
Thank
when
you.
we did this experiment, we did it with peripheral visual viewing expanded in stimulus. So when we do this, we expand this for peripheral vision bigger, no effect. Okay. So whether the peripheral vision make it expanded or not expanded to compensate for lower resolution peripheral vision, you still see the illusion regardless. The degree of illusion strength doesn't change whether you scale the whole stimulus up or not. Yeah. Thank you. This is
Okay,
a very
thank you.
important point to actually clarify. Thank you. Yeah. So,
No, thanks, Michael. Any other questions? I think we.
Kath, do you have and
Kath, what did you?
Yeah, can you hear me okay?
Yeah.
yes, yeah, I
Thank
can
you
hear
for a
you.
wonderful presentation,
Thank
Professor.
you.
I am not a vision scientist, so a lot of this is over my head, but it fascinates me the difference between central vision and peripheral vision. I'm a myopia control person, and we've had discussions around the differences between central vision and peripheral vision as a stimulus for myopia. So it's interesting.
Yeah, I'm very fascinated by your these days with the blurring of peripheral vision as a treatment. You wonder whether this can be kind of as a way of further studying, what is the mechanism to make it work?
Exactly. And how is that driving, how the eye grows during growth?
Yeah,
So
amazing.
beyond my capabilities, but very interesting to hear
Thank you. Yeah. And Peter, would you have some questions or comments? I'd love to hear from you.
I think Peter left. He had to
Peter
go
left,
somewhere
I
else.
see.
I think
Okay.
I see in the chat.
Okay. Yeah. And so, in fact, Michael, you make me realize that next time I should expand this figure because otherwise, yeah. So look at the whole, NEP feedback is already seeing that. You can see that this is maybe about, I don't know, 50 or 60 or 70 readout, and this is about 7. Yeah. And so therefore it's a factor of 10. Amazing. You can see a factor of 10 in terms of feed forward feedback, or no, in terms of feedback versus feed forward and relative to feed forward. It really could say that the peripheral visual field is qualitatively different from the central visual field. And this qualitative difference indeed is what we mean by dichotomy. It's central peripheral dichotomy. Of course, you can say it's a gradation. Yeah, it's actually a gradual thing. So therefore, dichotomy is a little bit of a you can say, a caricature. And so this, we need to know, this is actually gradually going down. Yeah. And so this is also something that could be further explored. So therefore, I would want to say that my framework perhaps opens more questions than giving answers. But nevertheless, it is a different kind of questions compared to the traditional study where we were, trying to understand what is vision and why things, this is the original question about, feature detector getting more and more complex. But now the new question needs to say we need to what is vision? Vision is actually overcoming the bottleneck and how to overcoming bottleneck. We are a successful species such that, even though we don't see the gorilla, we see the food in our foraging field. We see the dangers from predators such that we see the most important thing. And so therefore our looking mechanism is so successful to guide us successfully seeking the most important 1% of information into our survival. So this is quite amazing. And of course, in Chinese we say 杀鸡用牛刀. This is like you can use a sledgehammer to crack a nut. But if we can do a bit better, we can be more effective. So I wonder whether the future will really make us be very respectful to this species. Yeah. And if we understand this, we could build much clever, intelligent robots.
Perfect.
Yeah.
Good way to end. And on that note, I
Thank you
think
so much. Yeah.
brilliant. And it was really good to see the demos on a computer display.